It's my great pleasure today to introduce Chloe Blandino. Chloe is originally from Texas, where she developed a fascination with sharks as a child. She is a gifted artist, and for those of us who packed on a few pounds during the pandemic, she's also a certified personal trainer. And she received her Bachelor's of Science degree from the University of Hilo in 2017. She was also highly active in the Marine Options program there, doing a lot of scientific and community volunteering. I first got to know Chloe in 2016 when she actually came to HIMB as a volunteer for one of our HIMB alumnus, Dr. Mark Royer. And Mark worked his interns very, very hard. And I realized when I saw that anybody who could survive and thrive under that brutal regime would make an excellent graduate student. And so I encouraged Chloe to join the marine biology graduate program as a master's student, which she did. And she's been with us for 3 years. And today she will be presenting the results of her thesis research on the use of habitat by Sympatric sharks at French Frigate Shoals Atoll. Thank you.
All right, hi everyone. I'm just going to go ahead and screen share my presentation for you. Okay, Mark. Mark, how do I get back to my PowerPoint? Just says MBGP. Okay, found it. All right, awesome. Let me. All right, so I'm just going to dive right into this PowerPoint. In 1859, this English naturalist sparked widespread outrage when he suggested that our natural world was not created in just 6 days. He argued that the myriad of species inhabiting our planet actually arose from common ancestors that diversified through eons of struggle for existence. He arrived at this conclusion by observing the distributions of wildlife in fossils and the mechanisms influencing the distributions of organisms continue to intrigue ecologists today. For example, species with overlapping distributions may have to compete for resources. This can lead to competitive exclusion, where the dominant species excludes all others from the resource, or resource partitioning, where different species divide the resource to reduce interspecific competition among them. Closely related warbler species reduce interspecific competition among them for the same insect prey by foraging at different regions of the same tree. This is a classic example of spatial partitioning occurring among sympatric species. And sympatric species are species with overlapping geographical distributions. Now, terrestrial biologists have a distinct advantage when studying resource partitioning because they can directly observe the distributions and behaviors of terrestrial species like warblers with relative ease. Studying resource partitioning for elusive marine predators like sharks is far more difficult because these animals live in a concealing environment, making them fundamentally more difficult to observe. As a consequence, shark resource partitioning studies have heavily focused on diet because we can get this information by examining the stomach contents of dead sharks and by analyzing tissue samples. Although these studies are informative, they don't tell us very much information about important spatial and temporal associations between shark species, like how do shark species with similar diets reduce interspecific competition among them. Previous studies have attempted to characterize resource partitioning for Hawaii sharks by comparing the diets in the previous fishing locations of sharks caught in historic shark culls. Papastamatiu et al. Found that some species with similar diets exhibited broad-scale spatial segregation. Sandbar sharks were found to be relatively common in the main Hawaiian Islands and more rare in the northwestern Hawaiian Islands, whereas the opposite was true for gray reef sharks. A subsequent fishing-based study by Diel et al. Found that actually gray reef and sandbar sharks did co-occur in some habitats at French Frigate Shoals Atoll in the northwestern Hawaiian Islands, and generally broad spatial overlap among all shark species. So fishing surveys can provide us with snapshots of shark distributions, but they can also introduce sampling bias. Diel et al. Fished exclusively at day during the summer. So their results don't really tell us anything about shark distributions at night or during the winter. Furthermore, the bait that you use to attract sharks to your hooks may be attracting sharks from habitats that they typically use, skewing your understanding of shark habitat use. Thus, we need a fishery independent method of monitoring natural patterns of shark habitat use over long periods of time. And acoustic monitoring does just this. This method uses a uniquely identifiable ultrasonic transmitter that's implanted into sharks. And these transmitters are detected by acoustic receivers deployed at various locations across the seafloor. Acoustic receivers continually listen for these transmitters 24 hours a day, 365 days a year. And these transmitter tags can last multiple years. So this technique is particularly well suited for studying habitat use by sharks in remote settings like French Frigate Shoals Atoll, because acoustic receivers are continuously collecting data and they only need to be recovered and downloaded about once a year. I used acoustic monitoring data to evaluate shark resource partitioning at French Frigate Shoals Atoll. French Frigate Shoals is a classical atoll in the Papahanaumokuakea Marine National Monument, which consists of a 50-kilometer long barrier reef that partially encloses extensive sandy habitats, ribbon and patch reefs, and small sandy islets. Now, although these small sandy islets only account for a small percentage of the total land habitat at the atoll, they are critical nesting and haul-out sites to seabirds, sea turtles, and the critically endangered Hawaiian monk seal. So this concentrated biomass in organisms at sandy islets provide important foraging opportunities to sharks here. I compared the habitat use among the 3 most abundant species at French Frigate Shoals, including tiger sharks, Galapagos sharks, and gray reef sharks. There was a clear and significant large, medium to small size hierarchy at the atoll, with tiger sharks potentially serving as predators to gray reef sharks. Sharks were caught on fishing lines set in 189 locations across 3 broad habitat zones within the atoll, primarily during a single summer. These 3 broad habitat zones consisted of shallow lagoon and sandy islets, deep lagoon habitats, and reef habitats. A total of 129 sharks were detected by 24 acoustic receivers across a period of 2 years that were deployed at 14 acoustic receiver stations, which are represented by these circles, across the 3 broad habitat zones. I used a combination of shark size, habitat use, and diet to evaluate 3 different hypotheses about shark habitat use at the atoll. I predicted that small shark species would need to avoid overlap with large shark species to reduce predation risk. Shark species with similar diets should partition resources to reduce interspecific competition among them. And shark species with different diets would be able to exhibit a high degree of habitat use overlap because they wouldn't necessarily need to compete for the same prey. I quantified spatial overlap by applying a bipartite network analysis to the French Frigate Shoals shark detections. Network analysis links sharks, which are depicted by the different shapes, to receivers that they are detected on at any point in time across that 2-year monitoring period. Both sharks, both sharks and receivers are referred to as nodes in the network, and the links connecting them are referred to as edges. Node sizes, so you can see the different receiver sizes, reflect the frequency of shark detections. So the larger nodes equal more shark detections. Then I applied community detection algorithms that group sharks together with similar detection patterns across these receivers. So you can think of this as grouping sharks together with similar patterns of habitat use and core use area. Core use areas are habitats in which individual sharks are detected most frequently. In this figure, these groups of sharks, or in network analysis terms, communities, are represented by these colored bubbles. So the highest potential for competition or predation to occur among sharks is predicted to occur within these communities because these communities represent sharks with overlapping core use areas. The fast greedy algorithm identified 6 multi-specific communities. The colored polygons and the colored circles enclosing the habitat-coded receiver stations reflect these communities. For example, the red polygon represents community 1, and community 1 encloses 5 shallow lagoon habitats and 1 deep lagoon habitat. I found that tiger sharks were exclusively assigned to lagoonal communities. Gray reef sharks were primarily assigned to reef communities, and Galapagos sharks occurred in all communities. Now, it's important to remember that network analysis assigns communities based on core use areas. So theoretically, any individual across any of these species could swim across this atoll. But network analysis suggests that the core use areas, or the habitats that tiger sharks most intensively use, are lagoonal habitats, The core use areas for gray reef sharks are these peripheral forereef habitats, and Galapagos sharks have core use areas across all habitats. A similar pattern in shark habitat, similar pattern in shark habitat use was reflected in my residency analyses, which are calculated based on the number of days a single shark is detected on a single receiver, or otherwise known as site fidelity index. I found that tiger sharks have the highest site fidelity index in deep and lagoonal habitats, and the lowest site fidelity index in forereef habitats, whereas the opposite was true for gray reef sharks. Galapagos sharks have the highest site fidelity to deep lagoon habitats, but overall high site fidelity across all habitats. These spatial analyses indicate tiger sharks and gray reef sharks may be partitioning habitat, while Galapagos sharks exhibit extensive spatial overlap with both of these species. But these spatial analyses did not consider the possibility that interspecific temporal or time partitioning could also be occurring among these species. Temporal partitioning allows different shark species to reduce interspecific competition among them by utilizing the same habitats at different times. I quantified temporal overlap by comparing seasonal patterns and Diel patterns of shark habitat use at the atoll. I used non-parametric kernel density estimation of activities for all sharks to quantify seasonal overlap among shark species at every receiver station. And this approach calculates the probability that a shark will be detected at any point in time across a seasonal or a Diel cycle. This is an example of seasonal overlap occurring among 3 shark species at a forereef habitat. On the x-axis, we have the number of calendar months in a year, and on the y-axis, we have the probability that each of these species will occur at Rapture Reef. The peaks in activity reflect the intensity of use. So we can see here that Galapagos and gray reef sharks use Rapture Reef most intensively during the winter time, while tiger sharks exhibit more or less patternless behavior and whose lowest habitat use coincides with the peak activities of the other 2 species, implying that tiger sharks, or implying that forereef habitats may be peripheral habitats to tiger sharks. I found seasonal shifts between habitats for some species, and that Galapagos and gray reef sharks used forereef habitats most intensively during the winter, whereas both of these species used lagoonal habitats most intensively during summer. And once again, tiger sharks exhibited opposite temporal activity and were most active during the winter time. We're most active during the winter time. Now, I also found seasonal habitat shifts within habitat types. So in the previous example, we saw that tiger sharks were most active during the winter. And just 10 kilometers away in another shallow lagoon habitat, we can see that tiger sharks are most active during the summertime, and that there is no strong seasonal overlap among any of the shark species. So seasonal activity analyses up to this point have shown us examples of both seasonal overlap and seasonal partitioning occurring among species. But there is the possibility that species with high seasonal overlap may be partitioning habitat at finer temporal scales. So I examined diel or daily habitat use as well. This is an example of gray reef and Galapagos sharks using a deep lagoon habitat. On the x-axis, we have the number of hours in a given day, and on the y-axis, we have the probability that each of these species will occur at northeast of La Cruz. The peaks in activity reflect the intensity of use. So we can see here that gray reef and Galapagos sharks are most active at northeast of La Cruz during the daytime between 6 a.m. and 6 p.m. And both of these shark species exhibit overlapping diel habitat use at this deep lagoon habitat. Overall, all shark species were most active during the daytime in deep lagoon habitats and were most active during the day in coral reef habitats. However, diel activity patterns were far more variable in shallow lagoon habitats. For example, at trig shallows, Galapagos and gray reef sharks were most active during the day, whereas tiger sharks used this habitat most intensively during the night. Contrarily, tiger sharks were most active during the day at East Island, whereas Galapagos sharks lacked any clear diel pattern. And just 4 kilometers away in another shallow lagoon habitat, both tiger sharks and Galapagos sharks displayed overlapping nocturnal activity. So the big question here is whether the behavior exhibited by sharks at French Frigate Shoals Atoll support my hypotheses. I first predicted that small sharks would need to avoid overlap with large sharks to reduce predation risk. I found that tiger sharks were significantly larger than gray reef sharks, and previous studies have shown tiger sharks as predators to other shark species. Tiger sharks used lagoonal habitats more intensively than coral reef habitats, and in general, tiger sharks and gray reef sharks exhibited opposite seasonal habitat use and opposite spatial habitat use. And in the only network community in which both species occurred, tiger sharks and gray reef sharks exhibited completely opposite patterns of diel habitat use. These patterns are consistent with small shark species needing to avoid overlap with large shark species to reduce predation risk. And previous studies have shown how subordinate species will modify their habitat use in the presence of large tiger sharks. For example, in Western Australia, bottlenose dolphins will actively avoid their preferred habitat, foraging habitat, when tiger shark numbers are high. Now, the caveat to this is whether the behavior exhibited by sharks at French Frigate Shoals is primarily driven by preferred shark prey distribution. So we know that tiger sharks intensively use We know that tiger sharks hunt semi-terrestrial prey located at sandy islets, which would reasonably explain their intensity of use in lagoonal habitats. And although gray reef sharks primarily forage on coral reef fishes, which are widely distributed throughout the atoll, it could be the possibility that gray reef shark preferred prey are significantly more abundant in coral reef habitats. Secondly, I predicted that shark species with similar diets would need to partition resources in order to reduce interspecific competition among them. Previous studies have shown tiger sharks and large Galapagos sharks have overlapping diets, yet network and residency analyses provide evidence of high spatial overlap between them. However, I found evidence of both seasonal and Diel partitioning of habitat among these two species. Furthermore, there's also anecdotal evidence of each of these species specializing to different large prey to potentially reduce interspecific competition among them. This huge pulse in tiger shark activity at East Island during the summer and huge pulse in activity by tiger sharks at East Island during the morning corresponds exactly to fledgling albatross behavior. And although Galapagos sharks are known for predating birds in other regions of the world, only Galapagos sharks or only tiger sharks have been directly observed hunting albatross at French Frigate Shoals Atoll. Galapagos sharks most intensively use trig shallows during the summer and are most active during the daytime. This also corresponds with monk seal pupping behavior. And although tiger sharks could very easily take monk seal pups, only Galapagos sharks have been directly observed hunting them at French Frigate Shoals Atoll. Similar phenomena in other predator systems occurs as well. For example, at Yellowstone National Park, the reintroduction of gray wolves shifted the dietary focus of the subordinate cougar species off of elk and back onto deer. Thus, Galapagos and tiger sharks may be reducing interspecific competition among them by using a combination of temporal habitat partitioning and dietary compression. Now, the obvious response to this would be to collect more detailed data on the diets of each of these species, specifically at French Frigate Shoals Atoll. Lastly, I predicted that sharks with different diets would be able to exhibit a high degree of overlap because they wouldn't necessarily need to compete for the same prey. Among the 3 sharks I investigated at French Frigate Shoals, tiger sharks and gray reef sharks actually exhibited the lowest dietary overlap, but the strongest evidence for habitat partitioning. This may be a result of predator-prey relationships occurring between each of these species, and we know that predation risk is definitely a modifier in animal habitat use. The 2 shark species with the highest temporal and spatial overlap were gray reef and Galapagos sharks. And the diets of each of these species primarily consists of reef fishes. However, closer scrutiny at the diets of each of these species reveal relatively low dietary overlap at the family level. So of the 18 teleost fishes found in the stomach contents of Hawaii gray reef and Galapagos sharks, only 5 of those fishes co-occurred in the stomachs of both of them. And this apparent low dietary overlap among gray reef and Galapagos sharks would reasonably explain the high spatial habitat use overlap among both of these species. Other examples of low dietary overlap and high spatial overlap occurring among Sympatric species include Australian weasel sharks, whose diets primarily consist of cephalopods, while co-occurring carcharinids continue to eat fish. In summary, the habitat use patterns exhibited by sharks at French Frigate Shoals Atoll are consistent with prey distribution, predator avoidance, and resource partitioning. Tiger sharks may also occupy a keystone role at French Frigate Shoals because they are the apex predator here. So their prey preferences and habitat use patterns may influence those of other species. A keystone role for tiger sharks at French Frigate Shoals is further supported by trophodynamic modeling, where when tiger sharks were removed from the model, this resulted in the release of mesopredators and led to trophic cascade. Further research could clarify the ecological roles of sharks at French Frigate Shoals by surveying shark prey distribution in greater detail and by applying non-lethal molecular techniques to analyzing the diets and trophic positioning of sharks here. Of specific management concern is the potential increase in shark predation or predation risk to threatened and endangered species following the severe erosion of eastern Frigate Islands due to Hurricane Walaka in 2018. These sandy islets may no longer be suitable pupping and nesting habitats, so birds, turtles, and seals may be forced to relocate to other suitable habitats within the atoll. And of course, sharks who foraged at these habitats may also shift their habitat use patterns and prey focus in response to this. The highly specific habitat use patterns exhibited by sharks at sandy islets suggest that lagoonal habitats have unique characteristics in terms of shark prey availability. And the possible redistribution of seal pups or fledging birds may alter their vulnerability to shark predation. So, continued shark research, and I cannot stress this enough, is critically important to our understanding of the ecology of this fragile marine ecosystem. Thank you. First, I need to acknowledge the funders, data collectors, and field crew, those who lent advice for these project analyses and provided support to the fieldwork initiatives. Thank you. I want to thank my advisor, Dr. Carl Meyer, and my committee members, Dr. Lars Beider and Dr. Megan Donahue, for your tremendous support throughout the last 3 years. You've helped me evolve from the failure that my first project was to the resounding success that is this one. And I want you all to know that I stand on the shoulders of all 3 of you. Carl, thank you. This is awkward because you're in the same room with me, but thank you for being present and serving as a fatherly mentor to me. And I want you to know that I'm incredibly inspired by you, and I only want to continue to make you proud. And Dr. Kim Holland, who isn't pictured here, our shark grandfather, thank you for allowing me to do science on your turf. I thank these ground-shaking women that I am privileged to call teammates for literally getting down and dirty with me through the highs and the lows, and to all of our critically important interns who fished with me during all hours of the night with at least a partial smile on their faces. Thank you, Mark, for being an instrumental role to my academic trajectory, who took me in as his intern when he was a graduate student. And thank you, Fabian, for being a great marine mammal colleague and friend to me, and keeping me humble and reminding me that killer whales are actually the apex marine predators. I also appreciate all of the mentors throughout my life who helped me to ultimately get to the place that I am in now. Through the absolute best times and times I felt like my brain was going to physically vacate my body, I most thank my family, my grandparents, and my mom, who passed down a general appreciation of the sea to my brother and I. or a generational appreciation of the sea to my brother and I. And Victor, my partner in crime of 7 years, and his family for supporting me in any and every endeavor. My closest girlfriends who are also badass women in science. And this guy down here who gives me immense emotional support and who's the one guy that I love most in the world more than anyone else, my pup. Thank you. Okay, now I will take any questions. So I'm actually going to refer to the chat now, but thank you guys for tuning in.